Hi, everybody. Um, we are in this video going to go over the processes of spermatogenesis and oogenesis, fertilization, and growth and development. We will start with this process up here, steps 1 through 5 on the screen. We have a process that starts with 2 chromosomes in a cell and ends with 4 cells, each with 1/2 of that, 1 chromosome in each cell. And the diagram gives us a little giveaway. They've got little flagella or tails, so these are clearly making sperm. The process that makes sperm is called spermatogenesis, or simply meiosis. And this happens in the testes. We're going to start with diploid cells and end with 4 monoploid cells, each sperm, each monoploid. In that way, we're going to cut the pairs of homologous chromosomes apart so that where this cell at the beginning had 2 genes for every trait, the sperm only have 1 gene for every trait. And when we look down here, we've got a really similar process. We have, again, a cell that starts with 2 chromosomes but ends with 1 chromosome in each cell. Again, we are cutting the chromosome number in half. We're starting with meiosis, or in this case, because we're making an egg, oogenesis. Oogenesis, or the making of eggs by meiosis, happens in the ovaries. We're going to start with diploid cells where we have 2 genes for every single trait. We have pairs, and through meiosis, we're going to cut the chromosome number in half so that we have monoploid cells with 1 chromosome for each, but they're not all entirely the same. When we did spermatogenesis up at the top, we were making all 4 cells were sperm, and all 4 cells were monoploid. In oogenesis, we still have 4 cells. They're still all monoploid. But in this case, we have 1 cell that gets all the cytoplasm, and the other 3, number 9 here, all 3 are polar bodies. They're junk. We're going to get rid of those. And only keep the ovum, number 10, or egg, which is a monoploid cell, but it has all the cytoplasm, so that when this egg is fertilized, it'll have enough nutrients to survive until it can be supported in the mother's uterus. Again, all of these are monoploid. Now, what will happen is that sperm that was made by spermatogenesis and that egg that was made by oogenesis, they, of course, can come together. That process is called fertilization. And each of the sperm and the egg, as we said before, is monoploid. They were made by meiosis, so they have half the chromosomes. They are monoploid. Fertilization will fuse them and make a cell that is diploid. This cell is called a zygote, and it's diploid. Taking that cell and showing it again down here, we're going to show it growing into a full baby. So we're going to have the process of embryonic development starting with a zygote that is diploid, right? Because it had the monoploid sperm and the monoploid egg fusing together to make a diploid zygote. That is going to divide into 2 equal identical cells. And what process divides equal identical cells? Mitosis. And mitosis will occur again and again and again and again, a process that's called cleavage, until we get a whole bunch of cells all together that are identical to the original zygote, that have a copy of every single chromosome that came from dad and came from mom in the zygote, now copied during mitosis to grow into a full baby. At this point, we have an embryo, and every single cell is an exact copy of the original zygote. The zygote was diploid, so the embryo is all diploid. If this continues, we're going to have a baby, a fetus, and our full organism.